So, the next speaker is actually Katrina, the Institute for Global Food Security, School of Biological Sciences, Queen's University, Belfast, UK, and I see that Katrina is ready for action. Katrina, if you are ready, you have everybody's attention.
Hello everyone, and I'm very delighted to be here in Moscow and to present some of the research work I've been doing through a number of EU projects over the last 5 to 6 years. The title of my talk follows on very well from Professor4safety's presentation, because I am looking at marine and cyanobacterial toxin analysis and progress towards a novel bioanalytical supermice. I'm from Queen's University in Belfast, and on a nice day, our university is nice and bright with a beautiful blue sky, and on a bad day, we have similar rain and clouds to yourselves. We also have a lot of snow in Belfast from time to time, similar to Moscow, so there are some similarities. In relation to marine and cyanobacterial toxins, the different toxins are paralytic shellfish poison, such as saxitoxin and analogues, and these are produced by a species of harmful algae known as Alexandrium and other algal groups. They're particularly potent toxins. There's around 57 different analogues with all very much different toxicities. The next group of toxins are diuretic or lipophilic shellfish poisons, and their main group would be the ochideic acid and dinophysis, produced by Digotritis algae or Procenturium. The other groups of toxins that fall within that lipophilic group would be yesatoxin, pectinotoxin, and azosporacid. And then we also have amnesic shellfish poisoning toxins, such as domoic acid, produced by a species known as Pseudonypsia. 3 different groups of regulated toxins, both globally and within Europe. Very variable in chemical structure, very different physiochemical properties, diversity in toxin action, and also in their potency. Why do we test for these toxins? Well, these toxins either can make you very ill through vomiting, diarrhea, amnesic properties, and paralytic shellfish and paralytic properties. Basically, they have been named after the syndrome in which they cause. So paralytic causes paralysis and potential death, amnesic causes amnesia, diuretic causes diarrhea and vomiting. Normally these toxins are tested, or in the past these toxins have been tested using biological methods, and biological methods being the mouse bioassay. And there are 2 different assays, one for paralytic shellfish toxins and the other for the lipophilic toxins. So worldwide, through the 3 R's programs such as reduction, replacement and refinement for animal bioassays, there's a drive to progress towards scientifically non-animal procedures. So the animal assays were originally designed for acute toxicity as well. And there's increasing concerns now over toxin effects at lower levels and mixed toxicity. And food safety authorities recommending lower regulatory levels. There's also technical reasons in terms of sensitivity requirements for alternative methods. And to replace the mouse bioassay, analytical methods that can be fully validated to an international standard. So some of the alternative methods that are out there are functional assays, cell-based, receptor-based, enzyme-based, and there's a number of publications already presented on this type of work. Difficulties are there's low level of toxicity measured as relative to the toxicity of the sample, so that's a good point. They may detect new toxic analogues in structure. Toxin identification, though, is not unequivocal, so the assay will tell you that yes, you have a toxic sample, but it will not be able to tell you what that toxin is. And technology transfer of these particular methods is very difficult. In relation to analytical methods such as HPLC and mass spectrometry, again, there are methods already in place, And these methods are in place as screening methods as a reduction of the use of the mouse bioassay. Toxins can be identified and quantified for available analytical standards. Toxicity equivalent factors must be applied, and a sample cleanup and oxidation can be extensive for these types of methods. Data analysis is also laborious, but the mass spec method is unequivocal for toxin identification. However, they are expensive and they need trained personnel, usually for their use. So the methods that we focus on are biochemical assays, looking at ELISA and lateral flow devices and biosensor technology. Professor Dzamsky has talked about earlier. And these are binder assays, so toxicity does not always correlate with cross-reactivity. Sample preparation and data analysis, though, can be fast, and there's screening tools for hazard management, so for hazard analysis and critical control point management and rapid response. So types of screening tests then are based on antibodies, and basically we moved on from marine biotoxin testing because of chemical contaminants in food assays that had been previously developed for antibiotic residues, chlorophenicol, mycotoxins, and veterinary drug residues. And they're rapid, reliable, they can be low cost, no false positives, no false negatives, and are safe to use. And because food is produced on an ever-increasing scale, to enhance and ensure global food security, new biosensor technologies and applications are needed. So why are they needed? They're needed for speed, for higher throughput, in terms of lower cost. We're looking at portability. We're also looking for multiplexing, for remote sensing, sensing on site. We also want to meet the requirements of regulators and industry, and we really want rapid systems for end product release, so we can release food products quicker into the market that are known to be safe and free from any particular toxins. In terms of antibody production, we usually take our toxin, we conjugate it to our protein, we immunize our animals, and we end up with a polyclonal response. We also immunize mice, and within the mice there's an antibody response in the B cells. We take the B cells and we fuse these with myeloma cells and form an immortal hybridoma and produce monoclonal antibodies. And the best thing about monoclonal antibodies is that we have an infinite supply of antibody rather than just the supply from one animal. Some of the difficulties that we face in terms of toxins are the toxicity of the toxins themselves, that we don't actually kill the animal before we get an immune response. Also the source of the toxin, where do we receive some of these toxins to produce the antibody, and also the specificity, because you've seen in the previous slides some of these marine biotoxins have one parent, but maybe 57 different structural analogues of the same family of compounds. We have expertise in biosensor technology, starting off with ELISA and moving into lateral flow devices, and we've produced a number of different assays for marine biotoxins using both of these techniques. We've also used the BioCore technology, starting off with BioCore Q, moving to multiplexing BioCore, and we've evaluated a number of different biosensor technology platforms throughout. Going back to the BioCore technology, basically it works on the principle where you have your toxin immobilized. If you have no toxin in your sample, your antibody will bind to your surface. If there is toxin in the sample, less antibody will bind to your surface in a competitive manner. And in terms of the kinetics of the BioCore assay, you can see you will have a baseline, and when you inject your sample or your antibody over the chip, you will see it starts to associate. At the end of the injection, there's a natural dissociation, and we can regenerate the chip using acid or basic solutions back to the baseline. And that chip can be used over 4,000 times for some of the different toxins that we're analysing. And we get a calibration curve where when the concentration is low, we get a high response, to when the concentration is high, we get a low response. So in terms of the BioCore-Q, this was a single channel instrument, which really meant we could analyse one toxin family or group at a time, depending on the antibody. And we developed methods for amnesic shellfish poisoning, such as the domoic acid. We developed methods for diuretic shellfish poisoning toxins, for okadaic acid and the dinophysis groups. And very importantly was when we were designing these assays, we designed antibodies to match the toxicity of the particular toxins. We also then spent a lot more time developing antibodies to paralytic shellfish poisons because the potency of these particular toxins are lethal. So therefore, we had to make sure that we ended up with no false negatives because the difficulties then would be death, not just closure of beds for shellfish production. And again, we spent a lot of time developing antibodies to paralytic shellfish poisons. So we developed antibodies for saxitoxin antibody. One of the more potent toxins wasn't able to be detected. We developed antibodies to neosaxitoxin antibodies, and some of the other less potent toxins were not able to be detected, which was a good sign. And on a nice mixture of these two particular antibodies, we were able to get a mixture that matched the toxicity to the cross-reactivity profile of the antibodies. In terms of the multiplex analysis, then we had an instrument and the SPR real-time analysis, and on this one we had 16 different targets where we could immobilize on the chip. It could work as a 16:1 or a 4:4 format. So you can see here in the 4:4 format, we looked at saxitoxin, okadaic acid, domoic, and we also added polytoxin in as well. And instead of having one sensogram, we had 16 sensograms there within the one analysis. In terms of the extraction procedure, I go back to the physiochemical properties where we have lipophilic and we also have hydrophilic compounds in the one mix. But there's no point having a multiplex system if you need to develop different extraction procedures for your different toxins. So a lot of time is spent developing a single extraction procedure for shellfish. And in that simple extraction procedure, it was a matter of picking the right solvent and the right buffering system where you can actually extract and co-extract the different toxins simultaneously, quickly and efficiently in terms of a dilute and shoot approach. And the sensitivity of the antibodies that we developed allowed us to do that. So we were able to develop assays for saxitoxin, domoic acid, okadaic acid, and dinophysis, and polytoxin, whereby they met the regulatory limit, or they met limits that had been set, such as for polytoxin in other jurisdictions for shellfish analysis. But in terms of marine toxin detection, that was a lab-based instrument. However, it might be interesting to note we tested some algal and seawater samples that had been collected within a European project around Europe. And if you look at the analysis of these samples, you can see that in France, Italy, Sweden, Spain, Ireland, Norway, Scotland, and another area of Spain, the paralytic shellfish poisoning toxins are in the water. Albeit at low levels, they are present in the water. And the same for okadaic acid and some of the analogues. They're prevalent in a number of areas in a number of countries. And similarly for domoic acid, and this is in seawater samples. So what we set out to do was to develop a test that could be used as an early warning monitoring tool for water analysis. So we needed new tools for risk management on site to allow the industry, particularly the shellfish industry, to manage their harvesting and harvesting procedures. We also, within those studies, looked at cyanobacterial toxins, and these are also showing up in seawater samples as well. Microcystins and Slinders-Bromopsin, and currently there's no legislation. Microcystin has a legislation of 1 microgram per litre as a World Health, a WHO limit within drinking water. And again, you can see different analogues of microcystins and different structural properties, and then Slinders-Bromopsin as well. And these are known to be hepatotoxins leading to liver cancer potentially. So we decided, okay, we've got the 3 regulated marine toxins and we'll add these 2 to our assay to see what the prevalence is. So basically we had nanotechnology and microarrays. So we had printing, instrumentations, and we had a microarray cartridge. And on this microarray cartridge we can spot up to 80 to 100 different spots. We assemble it into this cartridge device. We drop on our solution or our water sample, and then we take a reading, and then in the reading we obtain our results. And the higher the fluorescence, the less toxin, and the lower the fluorescence, the more toxin. So again, this is the cartridge. So we have our slide, which we print here, our toxin protein conjugates. We also assemble it into a cartridge. And basically, the cartridge works on the principle of planar waveguide detection. So we have a diode laser, and this basically emits a beam and causes an evanescent field, and that evanescent field goes across our array spots. Our sample inlet, which is here and here, and we basically add our sample and it flows across the detection spots. And then there's a camera that measures the biomolecular interaction at that point. We basically then have a reader, and the reader we have nicknamed the toaster because it's as simple as putting bread into the toaster for toasting. And then there's a software package within the computer for all the data analysis and conversion and to allow signals to be sent. So in terms of the array, we basically spot the array. We've got microsystems, we've got pyrolytic shellfish toxins, we've got domoic acid, butyric acid, brevetoxin, polytoxin, slendersporoxin, and we're currently working still on the combination of these two assays into the array. It works on indirect competitive assay in the same way as the Bayes-Court assay, and this time we come in with a secondary fluorescent label to increase the sensitivity of the assay. And this is one of the examples for microcystin. So when we have no microcystin in the sample, you can see that there's nice bright spots, and when there is microcystin in the sample, spots darken. We looked at single analysis, so we looked at all the different toxins in their isolation, first of all, and we compared this then when we looked at the toxins all in the same assay. And basically, it was to make sure there was no real interference effects. There was a little bit of an interference effect with domoic acid, but you can see, other than that, they're very comparable in single assays and multi-assays. We also looked at running the assay as a 45-minute assay and reduced that down to 15 minutes. So we've got a 15-minute test that will measure the toxicity of 5 different compounds in water samples. And the sensitivity is very good in terms of low nanogram per mil to low picogram per mil for the more toxics within the group. And we're still working on some sample preparation for the water samples. We're also hoping to transfer this then into shellfish analysis and to allow then another tool to use for testing the shellfish meat. What we would hope is that this particular tool would be able to be used to monitor the toxin profiles should harmful algal blooms be appearing in the waters where shellfish are being harvested. So in terms of our multiplex assays, we had the Bayes-Court multiplex assay, which was a lab-based tool. We then have our array device, which is a portable sensing tool, and really it was to eliminate the use of mice for toxin testing. Really because we want to progress towards the development of an electronic mice for this type of testing as a measure of the 3 R's within European legislation, and to reduce and replace globally using a cost-effective diagnostic for the industry. Really, that type of diagnostic then we would like, as part of the Institute for Global Food Security, to transfer them to different commodities. So looking for mycotoxins in wheat and cereals, and also for then antibiotic or veterinary drug residue carryovers within feed, or mycotoxins within feed for animal health protection, looking at antibiotics in milk. When tankers come out to collect the milk from farm to farm can't be contaminated. Looking at shellfish meat within harvesting, water quality for different contaminants, industrial contaminants, and then different foods as well, because we think portable nanodiagnostics is the future for on-site testing to help ensure sustainability of food products. So just some acknowledgements to my colleague Stephen Queens and to Michael Lockhead from MBio, to the European Union for funding throughout for Bioconfidence, Midtown, Michael Aqua, and now Collab4safety. And just if anyone has the opportunity, we're hosting our own conference in Belfast, and even if you can't attend, we hope to upload abstracts and PDFs from that conference, and we'd like you to follow its progress. Thank you for listening.
Thank you very much, Katrina. For the sake of time.